Okay, one point that I haven't stressed and I should do is family history. So for the moment, a good family history, but I mean a good family history means 3 generations with your cousins and everyone included, provides a lot of information, but most doctors don't get a family history from you, yeah? So if you want, any family history is as valuable for the moment as predictive genetic testing most of the time. So you can do your own family history, even if your doctor doesn't do it. Go to this web page and they have a tool where you type in the things and then you can print nicely out your family history. And maybe you take it to your doctor and say, hey, look, this is my family. What do you think? Are we susceptible for this or that? You know? Genomics glossary. If I used a word that you have no idea what it means, go to this glossary. And so on. I'll send you this and you'll have the information. Now I will just talk very briefly about the microbiome and then go back to epigenetics. Let me just find which one it is. So I think it's this one. Yeah. So what's the microbiome? I had microbiology when I studied, and I didn't like all the bacteria and the bugs. I mean, I'm not a fan of microbes, yeah? But it's becoming more and more interesting. So I think I should learn more about bugs and bacteria. Why? There was a study, I think it was in New York, yeah, in New York, and they had little children who they measured, so they weighted and measured the little children when they were 3 years old and found that some of them were obese and some of them were normal weight. And then they got a lot of information from the parents and the doctors, and they found out that there was a group where only 15.7%, they had a group where 15% of the children were already obese by 3 years of age versus 7.5 in the other group. So what do you think is the factor explaining or behind that? And now I hear you. Bacteria. Which bacteria and how? The difference was the method through which the baby was born. So the babies that were born from cesarean section are the top group. The babies born by natural birth are the lower group. Probably going through the birth canal, that's the first bacteria the babies come in contact with. I mean, there's a flora in the vagina. And we still don't know what and which bacteria and so on, but apparently some of the bacteria that the baby comes in contact with while it's being born, and probably, you know, he also takes some and will swallow them. They go to his colon and so on. And they colonize. I mean, until that moment, his colon doesn't have any bacteria. So they're the first ones that get there. And now we know that all these bacteria that are living in our colon play a big role in metabolism. So there's people where they have, let's say, cleaned up all the bacteria from their colon and they got new bacteria through a fecal transplantation. That sounds a little bit gross maybe, but it's done and it helps some patients. And it makes a difference on their body weight also. So the bacteria that live in us, you can see here, I mean, we've talked about genome and genomics. We're just one being, yeah, with 20,000 genes. But our skin and our colon and mouth have a lot of organisms. And you can see the number of unique bacteria that we think are present in skin, for example, and in colon, and the number of genes that are encoded and found. So, I mean, there's many more genes if you add up all the bacteria in our colon than the genes that we have as humans. Yeah? So there's a lot of things going on that we ignored until now. Yeah? I don't know, I mean, that's what they proposed in this paper regarding body weight in 3-year-old children. It sounds plausible. But many studies are coming out that bacteria or microbiome plays a big role on our health, even on our body weight, for example. So the cells in our bodies are outnumbered 10-1 by microbes, okay? We have about 2 kilos of bacteria living in or on top of our body. So I think we shouldn't ignore them. I mean, they're there. They're doing something. So there's a big project going on. It's called the Microbiota project, where they are sequencing bacteria from different human tissues, from the armpit, from the mouth, from the nose, from the vagina, and so on, to see, and the colon, of course, to see what's going on. So experiments in mice have shown that genetically identical mice, if you change the bacteria in their colon, will lead to loss of weight. So this is the human microbiome project that's going on. And why didn't we know so much before, and what's the advantage of genome and sequencing for this type of analysis? Well, you can culture bacteria in the lab, but not all bacteria grow in the media that is normally used to grow bacteria. So many bacteria, we didn't know anything about them because we had never been able to have them grow in the lab. The advantage with sequencing is that you can take one bacteria from 1,000 bacteria around it and sequence its genome, and you still see, okay, I have one thing that is different from the other 999. And that's why studies like this weren't possible before. So all these studies are being made possible by the fact that we can sequence now the bacteria and then classify them according to different groups. And you can see that depending on which body part you look at, you have a different distribution of the type of bacteria. So now is the time to pay more attention to our bacteria. This is the Human Microbiome Project. And diseases that are postulated to be affected by microbes include all the ones that you see listed there: asthma, diabetes, stomach ulcers, and so on. This is a study that used this fecal transplantation. That means the patient has to, clean up his colon very well for a couple of days, and then it sounds bad, I know, but he gets a fresh aliquot of fecal matter from someone else injected into his colon. But there's patients that couldn't be cured by anything else. They had to try many antibiotic treatments because they had an infection with Clostridium difficile, and they couldn't get cured. And then with the transplant, it helped. So just that you know. Here you have resources. I won't say anything more about the microbiome. It was just, my idea today was just to open up maybe the horizons that you see all the applications and everything that's going on, and just decide what's relevant for you and read more about it. And now, do you still want to hear something? Or should I stop? No, should I go on a little bit? Okay. And then we'll have time for questions, which is a good idea. It was suggested. So let me see. So who's interested in your genome or your gene sequences? Now you see the picture. Takes a little bit. So science is interested, medicine, the pharmaco industry. If we have time, I'll talk about pharmacogenetics. So there's, about 20 drugs now on the market, where on the label it says that before you give that drug to a patient, you should test to see if they can metabolize that drug good or bad. Yeah, so there's drugs that help patients, and the same drug might even kill another patient. And it would be good to find out beforehand who's going to be helped by it and who's going to maybe have very adverse effects. Okay, China. I think they put China in there because there's this big Beijing Genome Sequencing Institute that is overtaking the sequencing market, and I think that's why they put China. Parents, marketers, insurance, law enforcement, employers. I put up this slide just to also show there's ethical and issues we could discuss about, you know. The United States has a law, it's called the GINA Act. I don't know what it stands for, but it was voted in the year 2007 where supposedly people are protected, even if they get the genetic sequence. Their employees, for example, don't have access and they cannot discriminate them by knowing this information and so on. Why would someone get genetic testing? Well, to see if they carry something, to do pre-implantation genetic testing. And I'll just mention this because I think it's something maybe it's not so known in our church, and I think maybe we should also think about it. Do we think it's ethical or not? So if a couple has difficulties getting a child, or if they know they both carry the predisposition for a certain disease, well, they can have in vitro fertilization, which is quite accepted in our church, I think. Well, the thing is that when you do in vitro fertilization, you have the cells until a stage of about 8 to 16 cells before they're implanted into the woman. And in that couple of days, you can take out one of the 8 cells, and it doesn't damage the future embryo at all. And you can do genetic testing on the 1 cell, and then find out if that possible future embryo carries the mutation or not. And then it's decided if you will implant that mutation or not. So it's kind of selecting for the healthy embryos, yeah? It is that. I mean, I have to say it. So it's something that our church should discuss. Maybe is it ethical, isn't it? I think it's also a very personal decision, but just that you know that's possible nowadays to do pre-implantation genetic testing. And then, of course, you can test the baby before it's born by amniocentesis testing and so on. Newborn screening. There's a lab in the United States that is offering testing for 700 genes. I mean, it's a lot. So it's a kind of many of the genes involved in Mendelian diseases that, you know, and they're doing that in like 5 days and for $600, just so that you have an idea of what's possible nowadays. And they're talking about sequencing the 10 genes that cause the most disease in children and future people, adults. Testing, they think they will get it down to $10 for sequencing 10 genes. So that you have an idea. I mean, this is not science fiction, so what's our stand on that and so on. And that's newborn screening, so after the baby is born, yeah? Then you can do pre-symptomatic testing, for example, to find out if you've inherited something which your relatives got as a disease, confirmed diagnosis, or forensic testing. Do you know that about like 200 people who had been in jail and some of them even died in the United States because of, because they also killed people there sometimes, were innocent, and it could be shown by DNA testing. So there's many people that have had an advantage from genetic testing that it could be demonstrated that they were in jail for something they hadn't done. So that's some of the applications. I thought I'll bring some more stories about how this is being used. So Nick Folkov was a little kid who had severe inflammatory bowel diseases for like 5 years, and he had more than 100 surgeries, and they didn't know what to do with this little poor kid. He couldn't eat any solid foods. He was really weak, and going from one operation to the next. And so in the end, they sequenced his genome and found that he had a mutation in a gene that had been previously linked to a blood disorder. So they decided to do a bone marrow transplant, and the kid is healthy. So if they, the doctors had no idea why he kept having this inflammatory bowel disease. And the child would have suffered another 100 of operations or died. But with the bone marrow, he's healthy. There's a very nice YouTube video about his mother and him and so on talking, if you want to look at it. Very interesting. These are the Berry twins on the top, and these are some other kids. So, this is not science fiction. It's being applied. The Berry kids had, I don't remember exactly. It was some kind of neurological disorder that had been well diagnosed, but actually the mother always had the feeling there must be something else. She knew there was something else, and I don't know how she got her twins' DNA sequence. And they found out that they actually had another mutation so they had the one that was known and expected, plus another one for which there was a drug. So now they got a second drug and are doing much better than they used to. So this is from another clinic showing you have a lot of children that are born with things which doctors cannot quite diagnose. And they've been able to find the mutant and the gene responsible for it in about 25% of the 250 cases that they've sequenced until now. And you might say, who cares? But it's important. It's important for the parents to know, do we carry the mutation, or was it a new mutation that arose just by chance that we don't have but our child has? So if we want to have more children, they don't run any risk. That's one point. Then it can help in the treatment. It can help parents to deal with the guilt that is sometimes associated with who passed on the mutation and so on. So there's a lot of applications. This is just to show you, for these rare diseases, they conducted this survey in the UK, and people just go from one doctor to the next doctor until they get finally diagnosed. Here you see it took 25% of the patients with rare diseases from 5 to 20 years to get their diagnosis. Yeah? And you can save them all this going from one doctor to the next by sequencing and finding out what's wrong. This is the number of doctors they had to see before getting their diagnosis. This is the best studied person there is until now. Professor Michael Snyder, he not only got his genome sequence, he also gave blood, I think, every 2 months for 2 years, and they followed a number of markers, and they studied his microbiome and everything. And when they did his genome sequencing, they found out that he had a predisposing mutation to diabetes. But Professor Snyder is very skinny. He doesn't have anything you would think he will develop diabetes type 2. So they just thought, okay, we found this, let's forget it. And they continued studying him for 2 years. And somewhere in the middle, he got a flu. He got fever, he was a couple of days sick in bed. And because they were testing him so regularly, they found out that actually he developed his diabetes after the flu. Yeah? So, I'm not saying everyone who gets a flu will develop diabetes, but in his case, it was very nice because they knew he had the predisposition. They thought, maybe false positive finding or who knows what. And then in the middle of the study, he gets the flu and he develops diabetes type 2. So this is just to show, I think, how medicine is going to change. Things are going to get much cheaper. We're going to be testing a lot of different markers and things more regularly to be able to be on the lookout for things that might arise. And then as soon as they arise, or even before, either prevent them or treat them as soon as they arise. This man wouldn't have gotten tested for diabetes for the next 10 years if he hadn't known this. So what's going on? You see all these nice centenarians. So these are all people older than 100 years. And there's a project going on where they got 100 people that are 100 or more years old, and they're sequencing their whole genomes. Why? Because probably sequencing these genomes of people who've lived so long will tell us or give us a clue, and comparing to people who've died earlier will probably tell us what genes and which mechanisms, which variants, not even mutations, which variants help us to live longer or function better and which ones don't. So just that you have an idea of what's going on. I've already talked about it. I'm not talking about this. So this is what I showed you before, this stick. And just that you have an idea, I mentioned before the genome-wide association studies, in short, GWAS. What you do is you take, let's say, 10,000 or 20,000 people who have hypertension, and you take 20,000 people who don't have hypertension. And it would be maybe still too expensive to sequence 40,000 genomes just for hypertension. So what you do is you just sequence little parts of the genome spread all over the chromosomes. So what you see on the left downside in different colors are the different chromosomes. And you just try to make a statistical relation between variants that are found in the hypertensive people versus the controls. And these are the dots that you see upwards from the line. And that means at those positions in those chromosomes, there are variants, there are changes in the letter of the hypertensive people that you don't find in the controls. And so this is all being used to calculate who is at risk and what's the risk for developing complex diseases like obesity. So is it easy to interpret what we find? At the moment, not so easy. Yeah, you find a change, and we're relying on some tools that we have, prediction and so on, but it's still, many times you're wondering, is this something that doesn't matter, or is it the mutation? But I think in the next 3 to 5 years, we'll have enough information that it will become much easier and it will be reliable. I'll skip this one. So genetic testing pros and cons. I mean, this is for a little bit of debate. Clearly, there are ethical issues involved. And as Viriato said today, you can use, I mean, every one of us has a knife at home, yeah? Even sharp knives. You can use knives as a good tool. You can use your same kitchen knife to kill someone, yeah? I think genetics is the same. I mean, you can use it for good, you can use it for bad, but we've accepted knives, so I think we might be able to accept genetics if we use it correctly. I think I'm going to skip this. I like this quote from Francis Collins. He said, he's the director of the NIH, yeah? And he said, we'll get closer to perfect kids from caring families and good education than we'll ever get from genetics. In case you're wondering, designer babies and whatever, yeah, you can choose the color of the eye of the baby if you really want to do that. It's possible nowadays. But I mean, the perfect kids, they don't come from the genes, they come from the family and all you give them. So now my question would be, do you want to listen a little bit more about epigenetics or about pharmacogenetics? Or you say, no, we don't want to hear anything else. So who wants to hear a little bit more about epigenetics? Can I see the hands? Okay, who wants to hear a little bit more about pharmacogenetics? Okay, we'll begin with epigenetics. Okay, so what we saw from Professor Kral, that I also mentioned, I mean, when monozygotic twins, identical twins are born, they have the same sequence, and at the beginning, I think even in the womb, they already don't have what's shown in this picture. So this turning on and off of the genes is usually illustrated by black and white signals. So the black ones are the turned off and the white ones are the turned on, yeah? So you see identical twins at the beginning, they have the same gene and the same gene is turned on and off. But as time goes by, you get different turning on and off of the genes, yeah? And like I said this morning, what you inherit is a book you get from your parents, but it has sticky notes. It has things saying, read me or ignore me. Okay, I talked about this morning in the panel. So this is this ENCODE project, and this was the main author of the study, and he's now facing a lot of criticism for stating that 80% of the genome is vital for life. So you can read this. I will skip it. I will skip this. I talked about mRNA at the beginning, and I think you understood when I wrapped up the paper and with a magnet, but in case you want to see it visually, this is the explanation. So you copy the DNA, you got the RNA strand, and it then goes to the cytoplasma where the protein should be made. And before the protein can be made, this little piece, this 25-letter piece, is swimming around there, either because your cell produced it, another cell, or nowadays gene therapy. And then it can stick to this blue chain and it will get destroyed before the protein is made. So I think everyone has asked or thought about in school, and so what makes the difference between a queen bee and the bees that have to do all the work? You know that genetically they're the same, so what's the difference? Well, there was an experiment that was made. You know that the queen bee gets a special diet. It's called the royal jelly, yeah? And what we have found out is that this royal jelly is very rich in something called DNMT3, which you see down there. What is this DNMT3? It's the enzyme. It's called DNase methyltransferase. So it's the enzyme that has to put the methylation signals on the different places. So apparently, this DNMT3 is more is somehow present in this royal jelly, and this leads to the bee developing to a queen bee. So they did an experiment where they injected, or I think it was in the food they gave to the bees, this RNA, this small RNA, to inhibit this DNMT3. And what they found out is that suddenly they had a lot of queen bees and almost no worker bees, just because they gave one 25-letter piece which inhibits this DNMT3, and that led to a complete reprogramming so that most of the bees became queens instead of worker bees. And that's the explanation behind the queen bee, yeah? But I mean, if you can see it so well graphically with the bees, I mean, think about us. I mean, we also have this DNMT3 enzyme in us, yeah? So what things turn it on and off? There's also a study that was done again by comparing children born by natural birth, VD, that's vaginal birth, and CS is cesarean section. And they compared at the time of birth in the cord blood, and after 5 days, the level of methylation going on in these two groups, and there was a difference. So the type of birth also influences the type of, or the amount of methylation that's going on this newborn child. I mean, I know it's cesarean section is necessary sometimes, but when it's not necessary, think about it twice before getting a cesarean section. I talked about epigenetics being able to predict age. This is the figure. So by testing or looking how many or how black, let's say, 5 genes are because they are turned off, you can calculate how old someone is. This is also, and as we age, and I showed that this morning, you lose our cells lose the ability to carry out this methylation correctly. So what you see here is the methylation in the inner circle, which you see like dark blue. That's a newborn, and you see all the chromosomes. So you see from chromosome, it starts on the middle, so at 1 o'clock, you see chromosome 1, 2, 3, 4, 5, and it's going on all the way around. And you see that in the newborn, most of the regions of the chromosomes are methylated. And as we age, on the outer circle, you see the methylation pattern of someone who's 100 years old or 103. And you see there's less methylation. So methylation plays a big role in aging, in cancer, in disease. So please eat a lot of things that help you methylate good, because you'll stay healthier and younger, really. So how is RNAi being used in the clinic? I guess this is still in development, but I think it will come to the clinic soon. So they had different mice, and they had them had an infarct. I don't know how, but they had them had an infarct. And then they treated the mice with this mRNA, this small thing, and they looked at the infarct size. So on the left side, you can see pictures, slices, so to say, of the heart, which probably most of you and I don't understand. But in the middle, you see the bars, and you can see the size of the infarct with the control, so the mice that had the infarct and didn't get any treatment. And then the gray and the black bars show you the size of the infarct of mice that got this mRNA injected right after the infarct, and how the size is smaller. So potentially there will be, I guess, in 5 years, maybe a therapy for cardiac things using miRNA. So I think I'll skip, well, I'll tell you just shortly. And I think it's important for women especially. So when you're pregnant, not only is your son or daughter developing, also the cells that will give rise to your grandchildren are also developing. So anything you do will have an effect even on the second generation. That's called a multi-generational effect. But if you really want to find out if the effect passes on from more, so it's not because maybe indirectly the cells were there when you, I don't know, had stress. You have to study in women the third generation. In men, it's enough to study the second generation. Yeah. So now you saw the agouti mouse, and if someone wants to Google that because he didn't understand what I was talking about today, it's written agouti. agouti. That's the name of these yellow fat mice, yeah? So they did another study to see if you could do something to contrast bad effects. And I don't know if I put the slides. Yeah, let me talk about this first before going back. So medicine has advanced a lot in the last years, but still, we see that there is an increase of various things. Even though we have better diagnostic methods, we have more therapies available. Breast cancer is on the rise. Prostate cancer is in the rise. Infertility is on the rise. So why? I mean, you have more tools, you have more medications, and still you're getting more cases. One explanation, I'm not saying it is the explanation, but one explanation may be something that we are all exposed to. Yeah, and there is a big lobby about this. It's a chemical compound. I think it's the most studied, or the chemical compound about which more papers have been written, about 5,000. And there's a lot of debate going on, and I think it's because there's a lot of economic interests behind this. So what am I talking about? Some may remember that around the year 2005, 2006, I guess Germany and Switzerland, I don't know the other countries, banned something called bisphenol A from being used in the plastics to make pacifiers and bottles for babies. But bisphenol A is being used everywhere, except in pacifiers and water and baby bottles, yeah? Bisphenol A is used in the lining of conserved cans. It's used in your PET bottles, so plastic bottles that you use to drink. It's used in plastic containers. It's used in the sealants that you get when you go to the dentist. And in the middle, you can see the increase in production, that means in the amount that's being used and we're being exposed to in the last 20 years, yeah? So there has been a study where they measure the concentration of bisphenol A in urine, and they found that the highest concentration is found in the urine of little kids, yeah? But from 2,500 people that they tested, 93% had bisphenol in their body that they were excreting. So I think all of us are exposed to BPA. BPA mimics estrogens. So it kind of fools the whole hormonal system, and that's why it has these maybe unwanted effects. It's been shown in experiments in rats and mice that it leads to the development of breast cancer. It also leads to a growth of the prostata and probably to prostata cancer. It has an effect on the brain, on weight gain, obesity epidemic. Yeah. Here you see, I mean, the bottle is just a depiction to see the concentrations. I guess you can't read much. Just look at the picture when you get it. I mean, we're being exposed to concentrations that in animal testings have shown effects about cancer, obesity, and so on. Yeah. So if you can, I mean, I didn't know anything about BPA until September last year when I was invited to give a talk, and I had to learn about teratogens, and that's how I came in contact with BPA. But since then, I've bought all glass containers to keep my food, and I've changed some things in my life. And let me see if I have it. Yeah. I told you BPA is actually not present in the sealant itself, but as soon as the dentist injects it into your mouth and the polymers start forming this chemical reaction, then BPA is produced in that reaction, yeah? And there was a study, it's not the only study, but I found this study really interesting. It was published last year in the Pediatrics Journal, which is very good. And in this experiment, well, it wasn't an experiment. They analyzed data that was available. They had children, I don't know in which country, who had amalgam fillings. I know amalgam has its problems because of mercury and everything, but anyway, there were children who got amalgam fillings, and there were children who got sealants that had BPA fillings, yeah? And they tested the children a couple of days later, and then I think they tested them like 5 years later, is it? I think they tested them 5 years later with different psychological tests, and they found a difference on the psychosocial behavior of the children that had gotten the BPA fillings versus the amalgam filling. So BPA filling led to changes in behavior in the children, yeah? So I'm not saying BPA is bad. I mean, on this paper, they say keep on using BPA if you want. And my recommendation for you is, I mean, I've read in a lot of other papers, it's been shown that if you clean your mouth with water for 30 seconds after the dentist puts in the sealant, then you reduce the concentration of the BPA to acceptable levels. So when you go to the dentist next time for a cavity, rinse your water, I think maybe better 1 minute, because I went to the dentist, I have a lot of cavities, and I did like this, I went, I mean, it was maybe 3.5 seconds. So rinse your mouth for at least 30 seconds to reduce the concentration of BPA. BPA is probably involved in a lot of things. So BPA also changes methylation and is involved in epigenetic changes. So now going back to what I wanted to show you like 5 minutes ago, what can we do about it? We're exposed to BPA. Well, they had these agouti mice, and then they had the agouti mice who were only exposed to BPA. They had agouti mouse who were exposed or received a control diet, and then the mice that were exposed to BPA but had a methyl donor diet. And as you can see, it's not perfect. And how you measure things in this agouti mouse model is you count the number of animals that are yellow, slightly mottled, and brown. Yeah, and that tells you how much epigenetic changes are taking place. And you can see that it's not that the BPA plus methyl donor diet kind of shifts everything in direction of the control diet. So even if you're exposed to BPA, and we're probably getting it from the can where we get our food and whatever, if you eat enough methyl donors, you can contrarrest a little bit the effect of BPA. Yeah? So just things that may not be so well known. This is a table that shows you not only broccoli, but also other natural compounds inhibit different epigenetic mechanisms that are involved in different diseases. So that's what you're seeing in the second, fourth, and third column. And yeah.
Does BPA enrich in the body? Does it store in the fat or what?
I mean, it stays. I mean, it's not eliminated right away. So these are just tables where you can see the role of epigenetics in foods. I don't expect you to read and know all this, but just that you have an idea. Oh, I didn't want to do that. And now I'll show you the experiment with alcohol. I wanted to go to that one. So what they did, they had, I think it was, I always mix of rats and mice. I think it was rats. And they exposed the pregnant female rat to alcohol while she was pregnant. Yeah, that's the first generation. And then for the rest of the experiment, no one else got in contact with any alcohol, okay? So when the pups were born, they measured the amount of pro-opiomelanocortin gene that was methylated. So it was turned off. If it's turned off, then you have less of the pro-opiomelanocortin protein, yeah, which is then broken up to make different endorphins and melanocortin and so on. So very key things for our normal functioning. And they saw that in the first generation, both the female and the male pups had different levels of methylation. So they had less pro-opiomelanocortin, both the male and the females of this rat that had been exposed to alcohol. Then, okay, I have 5 more minutes? 5 more minutes? Okay. So then they, so They had pups, this first generation with other animals that hadn't been exposed to alcohol. They had kids, and the same could be observed, but only in the males, yeah? And then a third generation, and even in the third generation, but only following like the father, the grandfather, there was this effect. So for males, at least in rats, it means if your great-grandmother on your father's side drank alcohol while she was pregnant, possibly you're expressing less pro-opiomelanocortin, yeah? The effect is different. It's only to the second generation on the female side. In epigenetics, we are seeing that many of the effects depend on female, male. Sometimes it's only on the male side, sometimes it's the other way around. So depending which gene, which agent, you have crossed effects, but there is a tendency to have a gender effect also. So I have to cut now because of the taping. So the ones of you who want to hear about pharmacogenetics can come back in 10 minutes, and the ones of you who have had enough can enjoy your time, and I thank you.